Okay, well, thanks for the invitation, and I really find this workshop really interesting. So putting it, thanks for putting it together. I'm going to, most of the talk is going to be focused on these ultra slow spontaneous sodium oscillations that we see in the neonatal brain. But if we have time, there is just a few slides on another topic that I'll briefly discuss that we just recently studied. So let me start with the main contributions to the work that I'm going to present. Carlos he's about to graduate as a PhD student, and Tatiana is a postdoc. They did most of the modeling. The experimental data on these slow oscillations came from Christine Roseley from University of Düsseldorf, and her PhD student did the experiments. And there's some data and modeling on the theta gamma amplitude coupling that I will present at the end of my presentation, which came from Elsa Tollner from Leiden University Medical Center in Netherlands. So just to quickly tell you why the spontaneous activity in the neonatal brain matters. And the reason is there is significant evidence by now that these slow spontaneous oscillations are spontaneous activity in general in the developing brain plays a huge role in cell maturation and their integration into complex brain region specific networks, and that's why we are interested in them. So I'll get right into the experimental data. Rose Lab can image, she can image sodium concentration in the neurons, so she can take neurons and astrocytes separately. So this side here is neurons. This is astrocytes. You can put them on top of each other to see where they are located in relation to each other. And the different colors of sodium dyes tell you which signal is coming from the neuron and which one is coming from the astrocyte. So if you select individual neurons and you follow the sodium concentration in these neurons, they basically show these fluctuations and spontaneous fluctuations with no aided stimulus. Same thing happens in astrocytes. They also show these spontaneous fluctuations, and this happens in the first week of the first neonatal week. After that, these oscillations disappear. So, for example, here the same experiment done during the third week, and you can see that majority of the neurons, they are really quiet. You see occasionally one or two spikes, but mostly both neurons and astrocytes are quiet in terms of their sodium concentration. So, and as I said, we do think they are important for the brain development in the early days. And they have really interesting properties. So they are ultra slow. They basically have a frequency about 2 spikes per hour. So if you look at the frequency histogram, for example, in the units of signals per hour, where signal means a single spike in the sodium concentration, the peak is around 1 or 2 spikes per hour. And occasionally you have more than 5, 6 spikes per hour, but mostly it's just really, really slow activity. And each spike can last for several minutes. So if you look at the duration of these individual spikes, there are some which will last for up to 20 minutes. Their amplitude is big. The sodium during each spike can change by as much as 7-8 millimolar. And you already saw that mostly these oscillations, they disappear after the first postnatal week. And another important thing is that not all neurons and astrocytes are involved. What that means is about 20 in the neonatal brain, about 25-26% of the neurons show these oscillations, and then most of them disappear in the later age. Similarly, astrocytes, about 40% of them show the spontaneous activity, and they disappear later.
Can I ask a quick clarification question?
Yeah.
You probably said it, but I must have missed it. Is this all over cortex, or is this in specific cortical areas?
So we image in CA1 region, but to be honest, I won't be surprised if they are in other hippocampal regions. Okay. So they are both in cortex and hippocampus. I think these are from the hippocampus area, these images, but they are both in hippocampus and cortex, yeah.
Okay, thank you.
Yeah. So, and another thing is that these spontaneous activity in the neonatal brain is observed in terms of calcium oscillations and in terms of burst of neuronal action potentials. But the difference with the sodium fluctuation is that calcium and neuronal action potential in the neonatal brain, they are really synchronized. They involve the entire network, which is not the case in our data. So, and they are not linked to, they are not linked to calcium oscillation. So here are some examples where we look at calcium in the astrocytes, for example, and then at the same time sodium in the same astrocytes. And you see that when you apply calcium buffers like BAPTA and EGTA and remove the calcium from the extracellular space, the calcium oscillation disappear, but the sodium oscillation persists. So they are not linked to the calcium oscillations. The reason I say this is because calcium and sodium in astrocytes specifically is really linked through different things, co-transporters and
Ghanim, may I ask you a question?
Sure.
So yes, but you apply BAPTA and calcium oscillations disappear, but it seems like sodium concentration oscillations became more pronounced and in amplitude and frequency and so on.
So this is actually one of the things that we are trying to figure out, that because there are so many things, so many transporters and so many co-transporters on the astrocytes that that could play a role in it. And the calcium channels and transporter depend, and the sodium ones, they depend on each other. So we really, to be honest, we really don't know why this happens. It could be just, for example, the sodium calcium exchanger depends on both sodium and calcium concentration. So if you change the calcium concentration is going to affect the sodium, the activity of the exchanger. So there are things that are independent, they are dependent on each other. But what we are trying to say here is that if you take the calcium oscillation away, the sodium oscillation don't go away. Does that answer your question?
Yes.
Okay.
Very cool.
Okay. So we tried to model it, and we basically started with a relatively simple network model, which is being usually used for gamma rhythms or theta rhythms, individual rhythms. We basically use 25 of excited T cells and 5 of fast spiking inhibiting neurons. And these cells are, and I'd like to mention that the number doesn't really matter if you scale the synaptic inputs, as long as you keep a similar ratio. You could use a smaller or bigger network to get the same results. And these cells are coupled through synapses and diffusion of ions in the extracellular space. So, ion concentration is important here. And astrocytes are not shown here, but they exchange ions with the extracellular space and buffer neurotransmitters like glutamate and GABA. The model, each cell basically have 3 key components. So the first component is the membrane potential, which is modeled by Hodgkin-Huxley formalism, and so we all know that I didn't include the, the individual currents, but we have the main current: sodium, potassium, chloride, and the pump. It also includes the potassium, sodium, and chloride concentration inside the cell in the extracellular compartment, and then as I said, the exchange with glia or astrocytes. And we also have the exchange of the diffusion of potassium or oxygen with the bath solution that is being used, or in vivo, that would be the exchange of oxygen and potassium with blood vessels. This component here is not, to be honest, not required to get the oscillations, but since I'll show you in a minute that a lot of things change during development, and we wanted to see, as much as we can, that what different pathways, what kind of role they play in this. So we include the release of the neurotransmitter, both glutamate and GABA, how it interacts post-synaptically, and its uptake through glutamate and GABA transporters. And that also is usually coupled with ion channels, transport of ion channels. So that is also included in the model. So when you put the network together, and there is no stimulus, everything just basically is normal the way you would model a developed network. You don't see any oscillations for obvious reason, because there is no stimulus, just like the experimental data. So then we said, okay, let's see what changes during the development. And there are many things that change during the development. So the big one is the GABA is depolarizing. In other words, in the developed brain, GABA transport usually brings in chloride to the cell, but in the first and second week, it's depolarizing. It takes chloride out of the cell. So that's the big one. The NMDA receptors, different types of NMDA receptor, their expression level, AMPA receptor, they all change during development. Sodium potassium ATPase also is really low in the beginning, and then it develops slowly by age, day 21, 22, it becomes almost 100% effective. The extracellular space is much larger in the developing brain. It's about 40% of the tissue volume as compared to 15% in the developed brain. The intracellular space is relatively small because these cells are still developing. Glia is not well developed. It means that it cannot buffer potassium efficiently. It cannot buffer the different different neurotransmitters. So we started, as I said, this is the big one. We started with that. We inverted the inhibition in the network, meaning that instead of the GABA synaptic, instead of bringing in chloride, it would take chloride out. And right away, you see these fluctuations in the network. With normal inhibition, there is nothing. With inverted inhibition, GABA inhibition, you see these slow oscillation, and they occur on the same time scale as you would see in the experiment.
What is?
Yeah. Was there a question? Okay. So another thing, I already mentioned this.
Question, if I can ask in between, or should I ask in the end?
You can ask now if you want.
Okay. So there was a slide where you mentioned your model with the voltage dynamics, so which was dependent on the pump dynamics.
Yeah.
So so in your earlier papers, so the pump dynamics was indirectly affecting the voltage dynamics through the ion currents, through the reversal potential of sodium and potassium. So I've only seen this paper, like the pump currents are directly depending on, or the voltage is actually depending on the pump. But in the earlier papers,
You mean,
it was not.
yeah, I think in maybe one or even two of the papers, there was a typo where we didn't write this in the voltage equation, but it should, because this is electrogenic, so it should depend on it should affect the voltage. It doesn't mean that we didn't include it in the model, but there was just a typo. We in the paper, we didn't write it. So we should correct that.
Okay, but even in the way at all paper, like the code was provided, and I have actually run that code, and which is without this I pump, and it's able to reproduce the results of the paper without this term.
Yeah. I mean, okay, so for the for the membrane dynamics, it doesn't play a huge role because it's very slow processes. So that's probably why it doesn't change much if you didn't include it in the membrane potential, but in the the effect of the pumps come in the slower ion concentration dynamics. So that's why it's more important to have it there. Although having said that, it should be included in the membrane potential equation as well, although the effect is not really significant.
Okay, thank you.
Yeah. Okay, so so as I said, these are not synchronous. If you take cells next to each other, several of them, they kind of oscillate independent of each other. But they are also there are no fluctuation in the extracellular potassium, and usually potassium and sodium are linked to each other because that's how the cell works. It absorbs sodium and releases potassium when it spikes. And so when you looked at the potassium, and it turns out that if you look at the single cell activity, you see small fluctuations, but nothing big like this. If you average the entire network activity, the potassium in the extracellular space still doesn't show much of the activity. Sodium, on the other hand, if you show the single cell activity, you see big fluctuations. If you average it, it disappears, and it's because they are not synchronous. They are just independent of each other. So and we know the reason why potassium doesn't fluctuate much. It's because the extracellular space is huge. So the potassium release due to spiking is not changing the extracellular potassium by too much. They disappear. These oscillations disappear when you block the voltage gated sodium channels, which is obvious. If you don't have voltage gated sodium channels, no sodium comes in, and the oscillation disappear. Glutamate doesn't seem to play any role in it, or just very little role in it. So if you block glutamate, it just not much changes. GABA, obviously, it's since we found that the GABA inversion is playing a big role. So if you block the GABA inhibition or the GABA neurotransmission, the oscillation go away. And the model does similar things. Basically, it follows what the experiments show. So there are things that you cannot test experimentally that we can check very easily in the model. So as I said, these cells are not very well developed. So we wanted to see if you have smaller cells versus larger cells, what happens. And it turns out that the smaller size of the cell supports fluctuations. They get bigger the smaller the cell becomes, and they get smaller when the cell grows. So here is a summary of that. The larger extracellular space, so here beta, beta is smaller. This is the ratio of it's the ratio of intracellular to extracellular space. So smaller beta means larger extracellular space and vice versa. It turns out that it's actually suppressing, it's going against the oscillation. So initially we thought that maybe the extracellular space would play a role, but it turns out that it's suppressing. So that's not helping the oscillation. And the glial buffering is also playing a role. So if you make the glial buffering weaker, you see the fluctuations. When you make it stronger, most of them disappear. So these things, this microenvironment plays a very active role during the development. Okay, so the network is very hyperactive in the developing brain, and this shouldn't be a surprise because the GABAergic signaling is depolarizing. Even if you have very normal potassium in the extracellular space, you see significant activity, and we believe that this is linked to the seizures that you know, where the developing brain is more prone to seizures as compared to the developed brain. Okay, so I want to switch gear and basically modify the same model and apply it to something a little different, which is a Dravet syndrome brain, and just show you some new results from that. Okay, so if you are not familiar with Dravet syndrome, what it is, it's basically a lifelong form of epilepsy that begins in the first year of life. It affects 1 out of every almost 16,000 individuals.
Regarding to our last session, the first speaker of the session is Professor Slatsky from my.
I think that I heard something in the background. Anyway, so they are characterized by frequent and prolonged seizures. There are behavioral and developmental delays associated with this condition. It's very high mortality rate, and it is basically a spectrum. It's the extreme case of the spectrum of disorders which are caused by mutations in the sodium, the voltage-gated sodium channels, specifically the type 1 voltage-gated sodium channels. And if you look at this spectrum, you start with the mild case. Usually, the mild case of these mutations lead to type 3 familiar migraines, and then it grows to febrile seizures where kids up to age 5 would have seizure, and then the seizures go away, and then there are cases where the seizure persists, all the way to generalized epilepsy, and Dravet syndrome is at the other extreme of this spectrum. So it's a important problem, and it's the issue with it is that not all people who have a mutation of sodium channel have Dravet syndrome. They don't go through seizure, not all of them. So it's kind of, a lot of them have this this mutation, but even some people who don't have the mutation, they show these behaviors. So we looked at the phase amplitude coupling in the mice Dravet syndrome mice model, both in the hippocampus and cortex, and it's impaired. And if you guys are not familiar with why the phase amplitude coupling matters, it's because it's supposed to be to play a very crucial role in the upper brain functions such as cognition and memory processes. So if we look at the local field potential from wild type mice, the raw signal here, the local field potential, and you filter it for theta frequencies between 3 or 4 to 14 hertz. And then you filter for the gamma frequency. You see there is a coupling, even from the time trace, you see a coupling between the phase of the theta frequency and the amplitude of the gamma frequency. And that's what we mean by phase amplitude coupling, that the amplitude kind of follows the phase of the theta. You do the same thing in Dravet syndrome model. the coupling kind of goes away up to a large extent. You don't see you don't see these clear peaks in the gamma rhythm. So and you can look at the power spectrum where your peaks are. They both basically in the disease model and the wild type model, the peaks occur at the same location. The power is very similar in the theta range and the low gamma range, which is basically 40 to 100 hertz, and the high gamma range in 100 hertz. And then you can also look at the commodulograms for the phase amplitude coupling, where you look at you take the phase of the theta and the frequency of the gamma, and this modulation index, it basically tells you that how strongly they are coupled to each other. So this is wild type. You see big bright spots at low gamma, and another bright spot at high gamma, and they disappear mostly in the disease model. And you can look at the average of these at low gamma, so both theta and low gamma. In wild type, it's higher, it drops significantly in the disease, and the theta and high gamma, it drops significantly. So we have started to model this. There are a lot of questions that we still are working on, but the basic model, basically, it works. We just had to make some changes. We basically, the excitatory or the pyramidal cell model, it remains the same. The fast spiking inhibiting neuron, the basket cell, they are very similar. And then we have the OLM, this other population of inhibiting neurons, which leads to the theta rhythms. So you put this model together, about the same network size, 25 excitatory, 5 each of basket cells and OLM cells. And what we did was we impaired the sodium conductance in the sodium channel in these inhibitory populations. Basically, the first question we wanted to ask is which one of the population is more effective. So in this simulation here, we inhibited both. We reduced the conductance of the sodium gated channels. And you see something really similar to what you see in what we saw in the experiment. So this is unfiltered signal, the theta rhythm, low gamma. You see kind of that coupling, and high gamma. You see there's some sort of coupling, and then some of it, actually most of it, disappears if you reduce the conductance of the sodium channel, or you make their gating slower, which could also happen in these mutations. And then the phase amplitude coupling again show very strong spots at the low gamma and high gamma, and it weakens in the disease case. So if you only impair the sodium channels on the OLM network, this is just the theta range here. The power shifts to the spectrum shifts to the left a little, and that's where this shift is coming from. But if you impair basket cells only, there is no shift. The power remains very similar to just like what we saw in the experiment. So this is basically the first indication that basket cells are playing the main role in this phase amplitude coupling impairment. You can also see how much you impair the sodium conductance or the sodium channels. So the stronger the impairment, the weaker the phase amplitude coupling, the modulation index decreases. If you impair the basket cells, are basket cells and OLM together, but with OLM only, you see something opposite. And again, that's the second indication that basket cells are the main population that are involved in this impairment. So there are a lot of things that we are still trying to understand and question that we are going to address. But coming back to the seizures, these Dravet syndrome mice are human, they show these chronic seizures, and they're really pronounced. Here's an example of EEG signal from the mice. They last for a while, and they usually appear if you. So let's say here, instead of using the Dravet syndrome mice, we ablated the voltage-gated sodium channels in the hippocampus, which is kind of a similar thing, but a local impairment of the sodium-gated channels in the hippocampus. And 7 or 8 days after the ablation, the seizure starts. So you see that a bunch of animals, and by almost 10, 11 days, almost 100% of them show spontaneous seizures. And the seizure number of seizures increases initially, and then it slowly drops to near zero with age. The phase amplitude coupling, it persistently basically stays normal in the wild type animals throughout the lifetime, although it does increase a little over with age. In the ablated the disease model, if you remove one copy of the gene or both copies of the gene of the voltage-gated sodium channel, you see a significant decrease in the phase amplitude coupling, and this is basically a summary of that. But what's important here is that by day 7, you see the decrease in phase amplitude coupling already, where the seizures start appearing later than that. And that's why we think that this impairment in phase amplitude coupling can be used as a proxy for telling if the Dravet syndrome patient is going to have seizures or not, because it's an indication that they usually the impairment starts before seizures occur. Another interesting result is that let's see, I can see my slide here. The impaired theta gamma phase amplitude coupling persists, and so the first slide, the seizure comes after the phase amplitude coupling impairment. This slide shows that if the animal is showing seizures, then the phase amplitude coupling would stay longer. The impairment would stay longer. Because not all Dravet syndrome patients are animals. They show phase amplitude coupling. Only a group of them show seizures, not phase amplitude coupling. But the ones that show seizures, the phase amplitude coupling. So here, the black one is the wild type animal. The phase amplitude coupling increases over time. And the red one is the animals, the Dravet syndrome model animal, but those that don't show any seizures. As you can see that the phase amplitude coupling recovers almost to the wild type level. But the ones that show the light gray one, the one that shows seizures, the phase amplitude coupling is significantly impaired even after 43 days. 42 days. And you can see this from these maps. In wild type, you see persistent increase in the phase amplitude coupling. This is the average over several animals. You see a slight increase, and that's because this also includes animals that do not show seizures, but the ones that do show seizures, they don't recover that well from the phase amplitude coupling impairment. So there are other information. The paper is in review, and hopefully it will come out soon. But there are still really interesting questions remaining in this problem, and we hope to address those in the future. So let me summarize what I said so far. Neurons and astrocytes in the neonatal brain show spontaneous ultra-slow sodium fluctuations They are asynchronous, are shown by only a fraction of cells, and are not linked to potassium or calcium oscillations. They are caused by depolarizing GABA neurotransmission and are ablated by blocking GABA and voltage-gated sodium channels. Glutamate doesn't seem to play a major role in it. Weak glial buffering and small intracellular volume favor sodium fluctuations, whereas large extracellular space suppress them. In terms of phase amplitude coupling, it is impaired in Dravet syndrome animals. It is mainly caused by the loss of function or mutation in voltage-gated sodium channels in basket cells. And then it precedes chronic seizures and persists in animals that show spontaneous seizures. I'm going to stop here. Thank you for listening. And if there are any questions, I'll be happy to answer them.